So from there, we're going to move to a little bit out of field. We're going to go off planet. And Monika Lipinska from HBBE at Newcastle University will talk to us about how we might be able to use nature to actually grow buildings.
Okay. My presentation is called Space to Grow, so I'll be trying to show you ideas of how we can use biological materials to grow habitats in space on Moon and Mars. I have a background in architecture and a background in space studies. And a couple of years ago, I was doing internships at NASA Ames Research Center with Lynn Rothschild at Astrobiology Lab, where I got a little bit of experience with working with biology, and I was bringing architecture expertise, which was an amazing experience where I was very free to ask questions because I didn't have any biological expertise. So that interaction with me bringing architecture expertise and me asking all of the simplest or maybe the stupidest questions, I think it was very fruitful. And from NASA Ames, my supervisor, Lynn Rothschild, she recommended my current supervisor at Hub for Biotechnology and Built Environment, where I get to continue the microarchitecture project. Just I had to find a parallel path, parallel approach to the project, so it counts as my PhD, like original content, but I'll explain that later. So currently I'm a PhD student at Hub for Biotechnology in Built Environment, which is an environment that allows me to continue connecting architecture with biotechnologies and biology, where I have supervisors coming from architecture and molecular biology. And the whole vision of this hub is to create buildings that are sustained by nature, sustained by life and life-sustaining, buildings that are living, which are responsive to the environment. They're grown using living engineered materials. They can metabolize their own waste, reduce pollution, generate energy. So the vision is very broad. What is really cool is that we have a lot of facilities like workshops and biological laboratories where even coming from architectural background, you can experiment and try to play in the lab with different materials and really have hands-on actual experiments. We also have this facility called OHM, which is experimental facility where we can test different materials, different samples that we're producing. It has like replaceable panels in the wall, so we can like actually create like wall structures, or inside there is a bathroom that we get to use, but also other people are running tests on like water recycling and microbiome. So it's very experimental and very like hands-on experimenting and playing with biology, basically. So the Hub for Biotechnology consists of 4 main themes. Building metabolism, that it's basically about processing waste and generating energy. So it's more chemical and biological on a smaller scale. Then there's living construction, which is about creating structural materials and having these living buildings. Microbial environments, that is focusing more on healthy microbiome and the interaction between humans and the environment that we live in and how the microbiome may impact human health. And then the last one is responsible interaction. So basically, how do we do all of these things ethically and responsibly and sustainably? Because I'm coming from architecture background and I'm working on a building architectural scale, I'm part of the living construction theme, where we're working with materials coming from plants like cellulose. So here are examples. This is actual bacterial cellulose, so it's cellulose produced by bacteria. And here are some examples of experimental projects happening in the hub where they were growing cellulose for facade material. And then the other project is actually growing cellulose by controlling the airflow. So unlike regular growth, which is by sheets, this one is more three-dimensional. So again, there's a lot of experimental work and just discovering and playing with materials to see what happens. Then we're working with fungi and specifically mycelium. So here are a couple of projects specifically using textile and mycelium. So there is a chair that my colleagues produce. It's knitted scaffolding that is filled with mycelium paste that dries out after mycelium grows. And it's pretty solid. You can actually sit on it and it doesn't break. Then there is another project called Bionet 1, because there's already Bionet 3 under construction right now. It's the same technology where you have scaffolding and you filled it with mycelium paste. What was really cool about this one is that it was actually grown upside down. So the structure was grown, it was hung in the like wooden scaffolding, basically filled with mycelium paste. And then after the mycelium grew and it dried and hardened, then they flipped it, and then we have this catenary dome shape structure. And then the last one is a Bionet 2, where again it's like a knitted structure, but then the mycelium paste is put on top of it. And after it dries, it also provides the structural support to the structure. So there is a lot of experiments, as you can see. And then we work also with algae. So I guess I don't have to tell you more about it. But there are some experiments with carbon, with tiles that can clean air and provide carbon neutral or carbon, I forgot the word that you were using on S. Sequestration? That's it. So we're working with that as well, and the idea is how you can print algae. And then finally, there are projects utilizing bacteria, for example, for creation of bricks, so microbiologically induced calcite precipitation, where bacteria are actually working as an agent, calcifying soil and making bricks out of it. My expertise and the projects that I'm working on, it's space architecture. So I'm trying to figure out how to use these biological materials to build off-planet. And then there are a lot of differences between planet Earth and then building on Mars or on the Moon or orbital stations. What I'm currently focusing on is the scarcity of resources. So here on Earth, we used to abandon resources. Now we don't have them abandoned as much as we used to, but in general, comparing to space, we can say that there are abundant resources. When we go to space, they're very limited. We cannot take that much with us because of the cost of payload and the transportation, and also we don't have that much on site. So the most popular concept is in situ resource utilization, so working with the materials that we find at destination, as we used to do on Earth with vernacular architecture. And then on the Mars and on the Moon, we would work with regolith, because that's the most abundant material, which is this loose layer of inorganic material covering the bedrock of the planet. And there are a couple of ideas how to use regolith to create structures, architectural structures. The thing is that the regolith itself doesn't have structural properties that can form architectural materials. It's basically like a loose soil. So we need ideas how to like 3D print it. We need extra nanopolymers or solar sintering to make it into construction materials. And here's where biotechnology or biomaterials can bring solutions. And I wanted to quickly show you two case studies with use of bacterial cellulose and mycelium, how we can utilize them to make structural components or structures in space. So the first one is bacterial cellulose, which is grown in sheets. And it has very great properties, especially for radiation protection. It actually has radiation protection properties that are better than water or hydrogen, which is very important when you build in space, because we have to protect humans against radiation. And here you can see how it grows, and then in the dried state, it's even stronger. It gets more properties when it's dry. So the idea was basically to reinforce regolith. So when you pile up loose sand, you have this angle of repose that the sand just falls, and then this idea is using principles of geotechnical engineering, where instead of the geotextile, like a layer of geotextile reinforcing the soil, we can use sheets of nanocellulose that it could be translucent, so it can introduce the sunlight into the habitat, but also it reinforces it so you can build vertically without additional materials, and also saving the footprint of the structure. And nanocellulose was actually tested, or cellulose produced by bacteria was tested on the surface of International Space Station on the EXPOS-R mission. And it's kind of similar to the melanin that we saw today. Bacteria producing cellulose are actually growing towards radiation, in a sense that the cellulose protects the bacteria. So they're producing cellulose to protect themselves. So it works pretty well in space environment. And here are some images how the project may look like, where the whole habitat is kind of like a sandwich structure, which is cellulose, regolith, and then towards the top of the structure, we would have more cellulose that provides radiation protection for astronauts, but also gives this human experience of experiencing the outside of the structure. And astronauts may want to, maybe they won't see the surrounding, but they will see the day and night changes, which are every 2 weeks. And yeah, so it's really important to think about these human factors. And I really believe that biotechnologies give a perfect tool for that. And also going to space as humans, we wouldn't be alone if we take biotechnologies with us, and they will help us. It will help us have our structures and live in space and also thrive in space, so we could live with them. I also have another case studies of mycelium, but I don't think I have time because I already have the red light here. So I guess I'll finish with that, and then I'll be happy to talk about the use of mycelium during the break and after the talks. And thank you so much.